Okay, my name is Bill Koven. I was in charge of the species, the gray mullet, and I'm going to talk about the objectives and the progress that we made. Okay, a little bit about the background and the rationale why we wanted to look at gray mullet. Gray mullet is a euryhaline, eurythermal fish found all over the world. In fact, it has a long history of being grown extensively. And we developed an IOLR diet that we're going to work with, which has no fish meal. It has some fish oil, which later on I'll explain that we can reduce that as well. It, I think, is unique that it offers an economical source of protein as well as a high-ended product of the eggs, which is called botarga. It's called karasumi in Japan. And at least I took a quick look at the internet, and I found it for 200 euros a kilo, although I saw this morning that it was 100 euros a kilo. And I think it's a fish for the future because if you look at the changing demographics in Europe, I think you're seeing that more and more people that have a tradition of eating mullet are living in Europe. So I think it is really a species for the future for European aquaculture. We started off with reproduction, and the problem with reproduction was that females do not progress past the early stages of vitellogenesis. They do not undergo final maturation and spawning. And also the problem of spermiating males are rarely observed, and in most cases the produced milk is highly viscous and fails to fertilize the eggs. So the objectives was to increase the abundance of spermiating males exhibiting high quality milk and to synchronize gonadal development in and between sexes and to induce spawning. So the hormonal treatment, which is the implementation of a hormonal therapy in this project at the onset of gametogenesis, successfully enhanced gonadal development. And it was a timed administration of recombinant FSH and an antagonist for dopamine D2 receptors at critical stages. And towards the completion of the reproductive cycle, a second hormone-based treatment consisting of GnRH and dopamine antagonist gave rise to successful spawning. So we had successful spawning, we had eggs, we had larvae. So just to summarize, the achievements in the reproductive part of the research is that they extended the natural spawning season to about 3 months. We shifted the spawning season, which can be easily achieved through photothermal manipulation. And they improved spawning success to about 60% and among hormonally induced females. We improved synchronization among breeding units, increasing fertilization rate to greater than 70%. And the relatively high fecundity, about 1.76 million eggs per kilogram, and the hatching rate was about 79%. And in 2017, we produced over 200,000 juveniles. Okay, so we have eggs, we have larvae. What are the questions that we wanted to tackle with the larvae? And one of the things we wanted to look at was the algae put into the tanks during larval culture. And this is done with all kinds of species. And the question was, what are the conditions? What is the concentration of the algae that we want to put in the tanks? And more importantly, is the turbidity it produces. When you add algae to the larval tanks, it produces a turbidity which the larvae can see their prey more clearly. Of course, if it's too little turbidity or too high turbidity, it's counterproductive. So we wanted to find what was the effective turbidity level. And we were also asking the question, why does algae work? Does it work because of this turbidity, or does it work because of some biochemical content of the algae itself? And here we looked at 2 levels of turbidity created by 2 different types of algae, Isochrysis and Nanochoropsis, against a control which had no algae at all. And we see that the higher level of turbidity, the larvae are eating more mastexes or meeting more rotifers, and so significantly more, so that suggests that they would be growing faster. Which is interesting here is that if you look at the survival at 51 days post-hatch fish, you see almost the same pattern. And where you have the higher levels of turbidity that the fish got during the first 12 days of life, and that they survive better. So what this does say is how important rotifer feeding is for future survival. Okay, well, we had the conditions for the algae that we wanted to add to the tanks. So the question was, can we replace this with clay producing the same turbidity? They do this in Norway with the halibut. Can we do the same thing with the mullet? And here we looked at 3 different types of treatments. One was the nano-B, the nanocoropsis at the high turbidity level. The control in this case was the low turbidity level caused by nanocoropsis. And then we compared it with clay, which produced the same turbidity. And as you can see, the rate of rotifer consumption was higher in the nano-B. The dry weight, the growth, was the highest in the nano-B as well, and it was significantly higher than clay, and also the same picture we saw with survival. So clearly, the turbidity was not the question. Apparently, it's the composition of the algae. So algae is fairly expensive. We're kind of very fortunate in Eilat because we have sunshine all year round, and that we produce a lot of live algae. But of course, to make it more economical, it'd be much better to produce a dry algae to feed to the tanks. You have off-the-shelf convenience, and it would be much easier and more economical. So we compared the live algae with a lyophilized substitute, a freeze-dried algae. And as you can see here, where you have the lines of nano, the dry algae, and nano, the live algae, the turbidity was the same. The consumption of rotifers, we counted mastexes, was the same. The swim bladder development was the same. Growth was the same. Survival was the same. So I think we can safely say that it is more convenient and economical to use the dry algae instead of live algae in the tanks. Okay, we also took a look at some of the enzymes that are produced from these algal treatments. In fact, we didn't see any effect of the algal treatments on the ontogeny of enzymes, but we did see some interesting things about some key enzymes such as amylase. As you know, all larvae start off as carnivores, but mullet at some stage becomes a herbivore or an omnivore. So we wanted to take a look at a key enzyme like amylase, which digests carbohydrate. And we saw that from 25 to 79 days post-hatch, the amylase actually increased 30 times. So it's saying that this fish is starting to change what diet it's going to be feeding on. And as it suggests a fish going from carnivorous to herbivorous or omnivorous. At this point, we didn't know which one it was, but something is drastically changing. So we also looked at it looked at 2 key enzymes to determine the maturation of the diet, and that was alkaline phosphatase, which is a marker for the brush border membrane, and leucine alanine aminopeptidase, which is a marker for intracellular digestion, which is the first type of digestion that a larvae goes through. When you look at the ratio, you can see with the higher the ratio, the more mature is the digestive tract, and we saw that the digestive tract matures at around 61 days post-hatch. What we also learned is that the enzymes from the brush border membrane and from the pancreas are pretty much genetically determined and not that much influenced by the diet. So this fish is basically predetermined to have this ontogeny of enzymes. And maybe it's not surprising because mullet in the open sea at some stage, as juveniles start to go to less saline waters, and such as the mouth of rivers and where the food and the salinity is very different. So in fact, it was not surprising that amylase, which is really characteristic of a fish that's going to become a herbivore or an omnivore, it would have to produce more amylase. Okay, now we came to the question, okay, we know when the fish starts to change its enzymatic ontogeny, but we want to wean the fish at around 25 days post-hatch. So do we feed it a carnivorous diet? Do we feed it an omnivorous diet? Do we feed it a herbivorous diet? We did an experiment. We actually used this product called Caviar, which was Bernaqua's weaning diet. It's from animal protein. It's about 60% protein. We used ulva that was grown at the lab, which is obviously high carbohydrate, low protein, and we used the combination of ulva and caviar at a 1-to-1 ratio. And we see here that survival, well, there was a tendency for the omnivorous diet to have the best performance, but the average fish dry weight was significantly better with the larvae that ate the omnivorous diet. And we even looked at the population, the weight distribution, and we came to the conclusion that the omnivorous diet resulted in significantly better survival, growth, and production of larger fish. Okay, we also looked at the enzymes at that point, and we see here that as you go from a high protein to a high protein, low carbohydrate diet to a low protein, high carbohydrate diet, the amylase does increase significantly. That's not so surprising. But we also saw the same pattern with alkaline protease and trypsin, and that was a bit surprising. And what we're suggesting is this is happening is because when the fish and the larvae are eating a source of food that is higher in carbohydrate and has amylase, it breaks down the carbohydrate and reveals more protein, which stimulates more enzyme production. We also saw that as you go from a less carbohydrate diet to a more carbohydrate diet, the alkaline phosphatase, this marker for brush border membrane maturation, goes down, and this other enzyme for intracellular digestion also goes down. So you see here, when you do the ratio, that the maturation index significantly goes down. So feeding a complete carbohydrate diet like ulva was not a good thing for the larvae. Okay, now we also want to take a look at taurine. And taurine, as you know, it's actually quite in fashion now. It's very important for bile salt production. It's very important for as an osmolyte, and it's important for muscle and neural function. And people have been realizing over the last few years that it's an important nutrient to take a look at. It comes only from animal sources. So if it's a strict herbivore, it will need taurine in the diet. We saw that if you look at, these are 3 different levels of taurine in the diet. At the rotifer stage, you see a significant effect at 6.4 milligrams of taurine per gram dry weight. And even after rotifer feeding has stopped, at 19 days, you still see the same effect. Here you see a similar effect that wasn't significant, but this was quite amazing. The fish that got this extra taurine just at the rotifer stage, we're still showing an advantage 32 days later after these taurine treatments have stopped. So this definitely is saying how important, again, rotifer feeding is and how important that taurine at early feeding contributes to larval quality. We looked at older fish, and we also found that there's a requirement for 0.5% of taurine in the diet. So if there's a requirement for taurine, then the fish has to be omnivorous because in nature it could only get it from an animal source. We also found, though, surprisingly, that the fish, the mullet, still has the capability of synthesizing taurine, and it synthesizes more taurine as you increase taurine in the diet. This also sounds counterintuitive. We didn't know how to interpret it exactly, but in the literature, there are cases where taurine is produced as an osmolyte, which maintains cell volume in the liver as you increase taurine in the diet. So we felt that this was what was happening here, and then you get to such a high level of taurine that you have a negative feedback system and it drops. Taurine is also very important for production of bile salt, and we see that this key enzyme here, this CYP7A1, converts taurine into bile salt, and it wasn't affected at all by taurine in the diet. So we feel that the ability to synthesize taurine here is also supplying any taurine that is required for the production of bile salt. Okay, then we looked at DHA in the larvae, and this was a bit surprising because we all know how important DHA is in larval rearing, but what we found is that our control, which was the low DHA, actually was red pepper, which has about 5.5% DHA, and that's the normal industrial standard. And we increased it to 12% DHA and 20% DHA, and we saw no difference in rotifer consumption, no difference in growth. We did see a difference in DHA in that the low level of DHA actually had significantly higher survival. So too much DHA in the case of mullet is not a good thing. We saw a very similar picture with juveniles, and that we had different DHA treatments ranging from 0.4% to 1.2%. There was no difference at all in wet weight gain or final dry weight, or even the weight distribution. So there seems to be no DHA dose-dependent effect on growth rate, final weight, and weight distribution. The dietary requirement is about 0.4% of the diet dry weight. Okay, now we come to something that we were thinking about for quite some time. These fish go from seawater to low saline of waters that are close to the mouth of rivers, and we wanted to find out how does this affect the DHA requirement. When you go from 40 to 8 or 10 or 15 ppt, does the DHA requirement, is it affected? And we basically, we expose fish to 3 different ranges of DHA because we change the DHA levels in Artemia and in this microdiet. And we see if those fish that were exposed to 15 ppt or 40 ppt, as you increase the level of DHA in the diet, you get a subsequent increase of DHA in the tissues. No, it doesn't make a difference if it's 15 or 40 ppt. But when we did this, we looked at older fish, we see that in the 40 ppt, you increase DHA in the diet, it increases in the tissue, but in the 15 ppt fish, there was no change at all. So this was the first hint that maybe as mullet go to a fresher water environment, a less salinity environment, it adapts the freshwater fish model, which has a capacity to synthesize long-chain polyunsaturated fatty acids. We also saw a similar picture with weight. As you increase the DHA in the diet, if the fish are in 40 ppt, as you increase DHA, there's a subsequent increase in weight. But again, in the 15 ppt, there was no change at all in weight. So it's another hint that the fish in lower salinity water has this potential to synthesize long-chain polyunsaturated fatty acids such as DHA. Okay, now we looked at the enzymes, and you know the marine, there's elongase and delta-6 desaturase, which are the main enzymes to make 18 carbon fatty acids into 20 and 22 carbon fatty acids. And in freshwater fish have this ability. to different extents, but they have this ability. Marine fish, by and large, they have these genes are not expressed. So what we saw, though, is that as you go from in 40 ppt fish, as you go through the different DHA levels in the diet, there was no change in the expression of this gene, this elongase gene. But if you looked at the 15 ppt fish, you see that the smallest level of DHA in the diet stimulated the highest level of gene expression of elongase, which makes sense. Why elongate fatty acids if you have sufficient fatty acids in the diet? You don't have sufficient fatty acids in the diet, DHA in the diet, then you're going to have higher stimulation of elongase. In desaturase, we didn't see that it was DHA affected, but we did see that the gene expression for this enzyme is significantly higher in the lower salinity than in the higher salinity diet. We also saw with SREP-P1 a very similar pattern to what you have with elongase. It's the gene expression is very sensitive to the level of DHA if it's at 15 ppt. So basically this is saying that as the fish moves towards lower salinity, as it does in nature, it has the ability to elongate and desaturate fatty acids in the diet to DHA and probably EPA and arachidonic acid, but we didn't test that. So the interesting thing here is that DHA can be reduced in the diet for the mullet if you grow the mullet at lower salinity. Okay. The effect of stocking density, we also tested in the growth of the fish, and we did find, and this is, I think, a major problem, is that as you increase the density, you're increasing larger numbers of smaller fish, and of course, that don't die. So, in fact, you're having an increasing number of fish that are eating but are not growing. And of course, this is the exact last thing you want in a fish that you want to monoculture. So we found this in ponds that are, or tanks that are polypropylene or cement. You can see here there's, as you go from concentration of, or at least a density of 2955 or 286 fish per square meter, you're getting higher levels of smaller fish. You see it here as well. The average weight decreases as you go, as the density increases, and also in weight distribution, so that the higher levels of, the higher density levels is encouraging the decrease of 3 minutes? Okay, I'm almost finished. And then when we tested it in Spain and in Israel and in Greece, we saw a similar pattern, but what was very clear is that there was, as you increase the density, you're producing higher levels of smaller fish. And in fact, this, I think, is a signal, is a warning that we have to work more on the grow-out diet because it's producing too many, it has a tendency as density increases that you produce higher levels of small fish. Okay, practical take-home messages for gray mullet culture. First of all, we improved the spawning performance, quality, and extended season through timed hormonal therapy. We know the concentration of the algae in the tanks, and that microalgae content and not turbidity is the dominant factor affecting larval performance. We can replace live microalgae with dry microalgae, not clay, and it's more economical. The omnivorous weaning diet was more effective in terms of gross survival production of larger fish, not a carnivorous or herbivorous diet. And improved spawning performance, quality, and extended season through, oops, I did that already. Taurine requirement was about 0.6% in larval feeding, about 0.5% in the juvenile feeding. Salinity is the driving force for DHA synthesis, and requirement during grow-out of juvenile mullet suggests lower levels of dietary DHA needed, which would be an economical thing because DHA is very expensive to put into diets. And as pond stocking density increases, the size distribution is skewed to smaller fish, whether in cement or earthen ponds or polypropylene tanks, and it's an obstacle to efficient monoculture. The entire juvenile production in 2017 was about 200,000 fish. Just publications that we've had, and that we have in preparation 9 papers. And this, I think you're going to talk about anyways. So here I'll just show you a quick video. One of our tanks is a 6,000-liter tank, and there's a few fish inside that. So that's it. Thanks. Is there time for questions?
Okay, thank you very much. Questions for mullet?
Baffled everyone.
Thank you. Do you think this thing about DHA requirement in freshwater is common for more than mullet or other species which are
Going from marine?
migrating, like salmon?
Well, it's the opposite with salmon. I mean, salmon go from freshwater to marine. But so their DHA requirement should increase.
Yeah, because I have this feeling that in salmon juveniles, you have they can make the DHA, but not so much in seawater. So that would be the same, but I'm not sure about that.
I don't know the efficiency of their ability to make DHA. I mean, you have trout. Trout has this ability to synthesize long-chain polyunsaturated fatty acids, but if you give preformed fatty acids, it's usually even more effective. So this still has to be looked into, how much we can reduce DHA in the diet and let the fish synthesize it instead, as long as it's at lower salinity water.
Bill, maybe I missed something, but you have shown relationships between the DHA level in the diet and the body DHA content. Mm-hmm. Did you show also the effect on growth, for example?
Yes, yes, it was the same thing.
It was the same, okay.
So that as you in 40 ppt, as you increase the dietary DHA, you see it in the tissues. But in 15 ppt, you don't see any difference. Okay. That was the first hint that maybe it's synthesizing so that it's not dependent on the DHA in the diet.
Just a comment that in the last Congress of Nutrition in Las Palmas, there was a presentation. Oh, sorry.
No, just
there was a presentation about the migrating flatfish. I don't know if you were there, and there was a Japanese presenting that. And it seems that this production of DHA in migrating fish is quite normal. So when they move from freshwater to marine water, they increase the production of DHA, their own production.
I mean,
so it seems that it's quite usual in migrating fish. Yeah.
It's not surprising because you're going from a marine environment, which is rich in DHA, rich in taurine. You're going to a freshwater environment, which is less rich in DHA and less rich in taurine. So you expect the fish to physiologically adapt to it.
I can see it's not surprising in the way you're saying, but I find it surprising that you've got a fish which I guess is capable of elongating to make the DHA, but when it's in a marine environment and it's deficient in DHA, it doesn't do it.
Why should it do it if it's an environment that has DHA? If you notice
but your diet was deficient, no? So it didn't have
it had
it.
low, medium, and high DHA. The diet that stimulated the highest biosynthesis of DHA was the low DHA diet.
Yeah.
And I think Mary Sowell also published something about that in Sebring, that if you give low DHA diet, it does stimulate some sort of biosynthesis of DHA. Probably not sufficiently, but that's the feedback mechanism.
But if I understood your graph, a deficient diet in the marine environment doesn't stimulate any production of DHA, but in the freshwater environment it does.
That's right.
So even though the fish needs more DHA, it doesn't produce it when it can.
No, because it's in a marine environment. Those are the fish that were in 40
why
PPT.
is that?
They cannot, they change their physiology.
That's the whole point I wanted to get across. I mean, you have to keep in mind, and I even suggested it with the enzymes, that the enzymes were genetically determined, even though you weren't really, not really affected that much by the diet. The fish in a 40 PPT environment is a seawater environment, and its physiology is sort of muted until it goes into, as far as biosynthesis is concerned, until it goes into a 15 PPT environment, and that stimulates this ability for biosynthesis. That's why I said it's the salinity is triggering this effect.
Yeah, but yeah, I agree. But I find it quite amazing.
Well, yes and no. I mean, in Israel, and I think in other places as well, it's known fact that they grow much faster in a low salinity environment.
The last studies we've done with the salinity, well, it depends on the capacity of the fish, of course. If they cannot, they cannot, if you change the salinity. But it seems that the salinity is very important for the enzymes, as you see. It's not a question only of temperature, not only for pH, but the desaturases are working better in the medium salinity. So if they are able to work, for example, if it's a marine fish, you move into 20, something like that, PPT, they can. For example, the Siganus canaliculatus or even the sole, but because they've got also a delta 4, another one. And the opposite, if you move one from 0, and if they've got the ability, until 10, something like that, you can also increase that. So it seems that the problem is even if they can, when they move to 35%, the salinity is affecting the capacity, so it becomes lower.
Yes, I mean, the paper that we want to write from this was actually the work by our master's student. It said salinity as the driving force.
Can I ask a question about the growth of the fish in the grow out? This poor growth that we got in 3 different experiments, in some more poor than others. How does this relate to the industry, where they're growing mullet in Egypt or in Israel using wild juveniles? What is the relative growth that they have in the same period? And how do they do it that they get so much different results? Because I'm sure they're not getting 100 grams in 18 months.
I think that, well, in Egypt, where they eat something like 200,000 tons of mullet a year, but it's all taken from the sea. They're all fingerlings that have been taken from the sea. And I think it's not grown intensively. And so I think, I'm not sure what diet they're using, but I think one of the problems with our diet is that, for instance, in a pond situation, the mullet are also eating the natural productivity. And I think that we have to have a diet that the fish their growth depends on that diet and not this sort of mixture.
You mean in the industry that they have also other things to eat in the ponds, not just what the feed that they supplement?
That's the situation at the moment. And I think, and I agree with you, this is sort of an Achilles heel right now, is that the grow-out diet has to be improved. I think we've shown that we get 20% plus survival up to juveniles, although I think we can improve on that, but I think what we have to concentrate now on is improving the grow-out diet. And that we're lacking something still that is a key, so that the fish will be attracted to the diet and not to the natural productivity.
You think it's the diet, it's not the husbandry, it's not the density, or it's not the pond versus both?
Yeah.
Not always. From what I understand, it's not always polyculture.
It's usually.
It's
I mean, we
usually
want to make the jump.
polyculture, so it's something that the behavior and maybe the microbial things that the probiotics, these could be affect their growth, because they do succeed to get a very nice growth rate, but in a very extensive manner.
I just want to make a comment that when you're growing these fish in higher and higher densities, and you get this sort of dichotomy of large fish and small fish, we did an experiment. I even have the results on one of the slides, is that we took small fish and medium fish and large fish and grew them separately. And the small fish actually had a significantly higher growth rate, and in about a month's time, they were all the same size. So really it's this interaction between individuals. And one of the things we want to look at is that we feel that there's some stress created, and that we may be able to modulate it with the diet by playing around with fatty acids and perhaps certain compounds that reduce certain eicosanoids. But that was one of the things we wanted to concentrate on. And you're absolutely right. It's an obstacle that has to be resolved. Otherwise, we're limited changing mullet from polyculture to monoculture. But I think it can be done.